Assalamu alaikum, Hamad. How are you?
Walaikum salam, ma'am. I am fine. And what's about you?
I'm also good. Okay, let me introduce you a little bit. My name is
Okay.
Arshia, and I have completed my master's in biochemistry and molecular biology and bring over 5 years of experience in tutoring biology
Mm,
at IGCSE and OA levels. My focus is on
mm,
concept clarity, structured exam preparation, and using proven techniques aligned with Cambridge standards to help students achieve top grades.
okay.
Before
Okay.
starting, before starting, I want you to know a little bit about you. Please introduce yourself.
Okay. My name is Hamad, and I am currently studying as AS level biology as private candidate under Cambridge International.
Okay, great. Do
Mm,
you like biology?
yes.
Okay. So, first of all, let me tell you about the strategy that how we are going throughout the session. In the first 4 months, I will cover the entire syllabus step by step, focus on concept clarity with short notes and diagrams for quick revision. And once the syllabus is complete, we will shift to assessment, past papers, and exam style questions to master Cambridge marking scheme. This way, you will have both strong understanding and excellent exam practice to score high with confidence. With confidence, okay. So
Okay.
this is the scheme we will be following throughout the session. And let me tell you the other thing that in my classes, I usually use interactive concept board, which makes learning very engaging. The best part of this board is that you
Mm,
will be having the full access to my board so that you can write or solve questions and do activities along with me, just like in a real classroom. Okay. Hamad, I'm sharing the
okay.
link of my chat, of my concept board in the chat. Just give me a moment. And what you have to do, you will just simply click on that link, and when you will click on that link, you will find the guest option. And when
Mm,
you click on that guest option, you will be able to join my board. I'm sending the link in the chat. Are you
okay.
able to see the link?
Yes.
Okay, now just click on that link and join the board. Meanwhile, I'm also side by side. I will also share the screen with you.
Okay.
Okay, now you have joined the board. Okay,
Okay.
so first of all, let me make you clear one thing that uh, although I will send this Word PDF at the end of the lecture, but I want you to
Okay.
note down important points. Okay?
Okay.
So,
Clear.
first of all, do you know that on daily basis that we take meals and we are taking nutrients, carbohydrates, proteins, fats in our diet, and so these nutrients are converted into ATP, energy-rich molecule. And every day the
Mm,
respiration process is going on in our body and other biochemical reactions are, you know very well, are taking place in our body. Do you agree
yes.
with this?
Yes.
So these reactions are basically taking place with the help of enzymes. In today's class, we will be studying about enzymes. Enzymes are basically the biological catalyst. Remember one thing, what are catalysts? Catalysts are basically, in chemistry, we use catalyst for the react to speed up the reactions. But why we
Yes.
use the term biological here? Because enzymes are biological catalyst because they are working in for the reactions taking place in living organisms. So that's why you use
Mm,
the term biological catalyst for them. They're basically the biological catalyst and they have specific shapes, precise 3D
okay.
structures. You know that they are made up of enzymes are basically made up of proteins.
Protein.
So the proteins have secondary structures, tertiary structure, and proteins are made up of amino acids. So because of the folding of these amino acids, they attain a precise 3D structure with hydrophilic R groups. And you know that they have specific active site for the substrate binding for to function, and they function both inside and outside the cell. You know that
Okay.
enzymes have basically in the reaction, there are three things. Enzyme is involved in the biochemical reaction, and the
Okay.
substrate on which the enzyme to which the enzyme bind.
Mm,
Basically, enzyme is having the specific active site. Or let me
okay.
draw it in front of you. For example, this is the enzyme, and this is
Mm,
the active site of the enzyme. In which active
okay.
site, for example, this is the substrate molecule. This substrate molecule will bind to that active site, which is complementary, that are complementary in shape only.
Okay.
And after that, ES complex will be formed, and ES complex will, the reaction will be taking place, and it will be converted into products.
Products.
So now we will discuss about the location of the enzyme. They can there are two types of enzymes are there. One is intracellular and extracellular. Intracellular are
Extracellular.
those, yeah, inside cells. Intracellular are synthesized and retained inside the cells. But for the extracellular, they're synthesized in the cell, but they are secreted out. And
Mm,
the function of both of them is the same. The speed of the reaction rate significantly, and the most important thing, students think that maybe this enzyme is utilized during the reaction, but this is not the actually enzyme. When the react Once the reaction is completed, the products are formed,
mm,
and you can again reutilize the enzyme for the other reaction. Enzyme can be reutilized. They cannot be utilized in the reaction. They remain unchanged
mm,
after catalysis for other reaction. Okay.
okay.
So, this is the whole enzyme action process. Enzyme and enzyme bind. I told you that substrate bind with enzyme's active site, and then ES complex will form, and then that ES complex, after when the reaction takes place, it can convert substrate into products, and the enzyme can
Okay.
again be reutilized. And the most important
Okay.
thing I want you, Hamad, to note it down. The most student confuse this thing that they think that enzyme uh, activates a reaction, but this is not actually the thing. Enzyme only reduces the activation energy required for initiating a reaction. Up till now,
Mm, okay.
everything is clear?
Clear.
Okay. Now, other thing is that there are two hypotheses for enzymes. The number one is lock and key, and the second one is induced fit hypothesis. For the
Yes.
lock and key, this enzymes, this model suggests that enzyme is like a lock, and the substrate is like a key that
Mm,
fits into it due to the enzyme specific tertiary structure.
okay.
Like enzyme active site is a fixed shape according to it is the enzyme active site is basically the fixed shape according to induced lock and key model. But what's
Okay.
the difference between the induced fit model? This hypothesis suggests in induced fit model that enzyme is like a glove and the substrate is like your hand. So induced fit is basically the one that it says that enzyme is the difference is that enzyme is not a active site is not a fixed shape. Its active site will be induced or slightly changes shape. When the enzyme substrate
Yes.
complex occur, it puts strains on the bonds and therefore lowers the activation energy.
Okay.
Now we will discuss about the factors affecting the enzyme. The first one is that we will discuss about the temperature. What will happen, you can see in the graph that when the temperature will be increasing, when the temperature is too low, the reaction rate is on the x-axis, it's about temperature, on the y-axis, it's telling us about the enzyme activity. So if the temperature
Okay.
is see that at 10 degrees Celsius and the temperature is too low, because of having insufficient energy, the reaction rate is low. But as soon as the temperature starts increasing, the it will gain the rate of reaction is increasing. until it reaches
Okay.
the optimum temperature. This, you know very well, the optimum temperature for human is around 40 degrees Celsius or 36 degrees Celsius. So up
Yes.
till now, there when the it reaches the optimal temperature, the rate increases. But what happened after that temperature? If we increase the temperature, if the temperature will be too high, we increase the temperature to 50 or 60 degrees Celsius. What will happen? It
Mm,
in high at high temperature, the bonds between the protein between the amino acids will break down, and when
mm,
the bonds will be break down, it will lose its tertiary structure. So definitely when the it will loses its specificity, it will lose its active site shape, it will not be able to bind to the substrate, and in this way, that's why the reaction rate will start decreasing. now the other
okay.
Now the other one is about the pH. What happened? Enzyme will work only at the optimum pH, even not at too high or too low pH. What happened when the pH will be too high? You know very well that pH means actually the hydrogen ion concentration. So when the hydrogen ion
Yes.
concentration is too much high, the enzyme, these they will interact with the amino acid. You know, amino acids are having charges on them, positive and negative charges. So due to the
Yes.
high hydrogen ion concentration, the charges on the amino acids will change. So definitely the bond between, they will not be able to retain the bonds between them, and when the bonds break, they will lose their active site, or they will lose again their structure. So that's why the enzymes also work on optimal pH, not too low or
Okay.
not too high, not in alkaline environment or acidic environment. So here
Okay.
are the key summary points that we have discussed today. Number one is that enzymes are specific protein catalysis, that they
Mm,
only lower the activation energy. We have discussed about lock and key model and induced rate model, and how the temperature
okay.
and pH is affecting the enzyme structure.
Okay.
So up till now, everything is clear. Should we move towards the assessment?
Yes, ma'am. Clear.
Okay. So are you able to see clearly the board? So the first question is, which statement about enzyme action is correct? Enzyme raise the activation energy of reactions. Enzymes are consumed during the reaction. Enzymes lower the activation energy of reactions. Or enzyme change the equilibrium position of reaction. Think about it and tell me what will be the right answer.
Option C.
Good. Now come to the second
Enzyme lower
one.
the activation energy.
Yes, good. Now
Okay.
the second question. The optimum temperature for human enzymes is approximately 20 degrees Celsius, 37 degrees Celsius, 60 or 80 degrees Celsius. Which one will be the right answer?
Mm, wait, ma'am. B.
Yes, 37 degrees Celsius.
Okay.
Okay, now this was your then we have done about the assessment MCQs, and in the last, I always add one assignment question for the students, which will be your assignment. So this is from your past paper question, that you have to let
Okay.
me guide you a little bit, that you have to explain why enzyme activity decreases at high temperature even though the molecular movement increases. Include reference to protein structure in your answer. And what I'm expecting from you for these 6 marks, that you have to discuss about denaturation, hydrogen bonds breaking, active site shape change, loss of complementary fit, and irreversible process. Okay, so
Okay.
this is your assignment, and tell me that everything will be clear, or you do have
Yes,
any
ma'am. Clear.
question? Okay. So this is end for today's session. Thank you so much for your time.
Okay.
Allah Hafiz.
Hafiz.